Welcome to the next episode of the microbiology tube. So today we'll be talking about the bacterial conjugations. So bacterial conjugations is the process by which one DNA is transferred from one bacteria to the another bacteria by the help of the sex pillars. All the bacteria are not capable of having the procedure called the bacterial conjugations because all the bacteria do not have the F plasmids. So only those bacteria who have the F plasmids can transmit the one DNA from one bacteria to the another bacteria. So the transformation is the process by which the naked DNAs are transferred from one bacteria to the another bacteria, and no any mediators are required. And in the transduction, the mediator for the transfer of the DNA from one bacteria to the another bacteria is the bacteriophage. But in the conjugation, the DNAs are transferred from one bacteria to another bacteria by the sex pillars. So in the 1947, so Landerbach and the Tautum, so these two has performed an experiment. So what they did is that they have the two strands. So they have the two strands, the strands A and the strands B. So strands A is unable to make the methionine and the biotin. Similarly, strands B is capable of producing the methionine and biotins. So what they did is that they put this strands A into the media which contains the minimum nutrient, and what you can see is that there was no growth. Similarly, he put the strands B in the minimal medium, so there was a minimum amount of nutrients, and you can see there was no growth of the bacteria. So but what they did after that is that so strands A is mixed with the strands B in a test tube. So they left it for some time so that there may be the conjugations. So and what they did is that they placed that bacteria again into the minimal media, and you can see there is the growth of the bacteria. So at first strands B was unable to synthesize the methionine and the biotin, and similarly the strands B was unable to synthesize the threonine, leucine and the thionine. So but at the end, if there was the two bacteria were mixed, what they did is that they transferred the one DNA from one bacteria to the another bacteria, and you can see some of the bacteria have survived in the minimal medium. So those bacteria which have survived in the bacterial medium, so these are the bacteria in which conjugations was performed or the DNA was transferred from one bacteria to the another bacteria. So what they did is that, so I have already talked you about the sex pillars. So this is the one bacteria and this is the next bacteria. So what they do is that they extend the sex pillars, this is the sex pillars, and they transfer the DNA from one bacteria to the another bacteria. So this is the photographs taken by the electron microscopy. So first, what is the plasmid? So as we know, the plasmids are the small double stranded molecules which are independent of the separate replications and have their own replications origins. So plasmids are the episomes. It means the plasmids can exist with or without being integrated into the host chromosomes. So they can integrate into the host chromosomes or they can be independent. So the best study is the conjugative plasmids. So conjugative plasmids is the F plasmids I have already discussed earlier. So if you see here, this is the F factor or the F plasmids. So the F plasmids are the these are the 1 lakh base pairs. So these are about 1 lakh base pairs long and bears genes responsible for the cell attachment and the plasmid transfer between the specific E. coli cells. So you can see here there are a lot of the genes which are responsible for secreting the enzymes needed for the conjugations, whereas also the enzymes needed for the developing of the sex pillars. So there are the different types of the cells involved in the conjugations. One is the F plus. So there is the F plus cells, F minus cells, and F prime cells, and the HFR cells, which is called the high frequency recombinant cells. So you can see, first I will discuss about the S plus cells. In the S plus cells there will be F plasmids. So this is also called the male bacteria. So in case of the F minus cells, in the F minus cells there is not the presence of the F plasmid, so this is also called the female bacteria. So you can also call it as a male bacteria, and it also called as the female bacteria. So So this has the F plasmids, but this F minus doesn't have the F plasmids. So there is next type of the bacteria which is called the HFR, high frequency recombinant cells. So you can see here in the HFR, these are the bacterial chromosomes. These are the bacterial chromosomes. So these are the bacterial chromosomes, and this red part is the F plasmids. So this red part is the F plasmids. So in some of the bacteria, so the F plasmids get integrated in the bacterial chromosomes, and this bacteria is called the HFR, high frequency recombinant cells. So next is the F prime cells. So what happens is that after the formations of the HFR, some cells have capacity, some cells or some bacteria have the capacity in which the DNAs, the integrated DNA becomes separated. So during the separations, what happens is that not only the F plasmids will be separated. So in some cases only the F plasmids can be separated, but in some other cases F plasmids along with the bacterial chromosomes will go. For example, this is the bacterial chromosomes, and here you can see this blue color, this is the bacterial chromosomes, and this red part is the F plasmids. So you can see the separations of the F plasmids along with the bacterial chromosomes, and this stage is called the F prime cells. So I repeat, so the bacteria which have the F plasmid is called the F plus cells. The bacteria which do not have the F plasmid is called F minus cells. Those bacteria in which the F plasmid is integrated with the bacterial chromosomes is called the HFR cells, and those bacteria in which after the combinations of the S plus, again the S plus is separated along with the bacterial chromosomes, that is called the F prime cells. So first I will discuss you about the matting of the F plus and the F minus. So you are very clear with the F plus. This F plus means the bacteria which have the F factor or the F plasmids. So in the diagram you can see this is the F plus. So here is written this is the F plus, and F plus is the male bacteria, and as well as it is the donor cells. So you can see here the next is the recipient cells. So this is the F minus cells. So this is your F plus cells, and this is your F minus cells. So F minus cells is also called the female cells. So what happens is that the F plus cells will make the sex pillars. So this is the sex pillars, and this is the sex pillars, and that will generate the sex pillars, and that will attach to the F minus cells. After the attachment by the sex pillars, they will contract. The two bacterial cell will contract, and after the contraction, the F factor from one bacteria will be transferred to the next bacteria. It means the F plasmids from F plus cells will be transmitted to the F minus cells, and after the transmit of the bacteria from F plus to the F minus cells, the F minus cells becomes what? The F minus cells becomes the F plus, because at the initial condition the F minus cell do not have any F factor, but after the conjugation the F minus cell consists of the F factor, so it becomes the F plus, and two bacteria becomes the F plus. So and they get separated. So this is the matting of the F plus and the F minus cells. So matting of the F plus and the F minus cells will change the F minus into the F plus. That will change into the F minus into the F plus. So next is the matting of the HFR and the H minus. So as we know, the HFR stands for the high frequency or recombinant. So this is the high frequency recombinant cells. So if you see here in the F plus and the F minus matting, so there is not the recombinant. So the recombinant is very small, into 10 to the power 3 to 10 to the power 4 bacterial cell in bacterial cell. So only one probability is there the bacteria can be recombinant. Recombinant means these F bacteria will attach to the bacterial chromosome, so that is called the recombinant. So there is less chances of becoming the recombinant, but for becoming the F plus, there is 100% chance of becoming the F plus if the F factor enters into the bacteria, but there is the less chances of becoming the recombinant. But in case of this HFR, so HFR means high frequency recombinant, so it means that there is higher probability of becoming the recombinant. So any bacteria can be recombinant at higher frequency, so this is called HFR. So if you see here, this is the F plus cells. At first this was the F plus cells, and actually what happens is that the part of this, what happens is that when this these plasmids get integrated into the bacterial chromosomes, this was called the HFR cells. So as I've already talked to you in the previous slides, so initiations of the F factors, initiation of the F factors into the chromosomes. So here you can see here, this is the HFR cells, and in the HFR cells, and there is the next bacteria, which is the F minus cells. So F minus cells are the female cells, which is the recipient, and HFR will be the donor cells. So what actually happens is that the HFR will transmit the F minus cells. So in the transfer of the chromosomals, in the transfer of the DNA, so only the small portion of the F plasmids will be transmitted, but the larger portion of the bacterial chromosomes will be transmitted in this step. So it means in the earlier step, in the F plus and the F minus steps, only the F plasmids will transmit, but in the matching of HFR and the F minus, so what actually happens is that a small amount of the S plasmid will transmit along with the larger bacterial chromosomes. So small F plasmid will be transmit, and larger bacterial chromosomes will be transmitted. So you can see here, so from here you can see this red indicates the bacterial plasmids. So this is the plasmids, and this blue represents is the bacterial chromosome. So you can see the larger portion of the bacterial chromosomes is there. And what happens is that these bacterial chromosomes and these larger bacterial chromosomes will be integrated into the F minus cells, and this will become the HFR. So HFR and H minus will result in the formations of F minus into the HFR. So similarly you can see here, so if it takes a shorter time, so only one gene can be transmitted. So for example, the lactose producing gene can be transmitted, but if it takes a longer time, then many genes can be transmitted. For example, lactose or the proline genes may be transmitted. So this is what it is showing here. So if we kept the HFR cells to mate with the F minus cells, then the formations will be the HFR cells, that is the high frequency recombinant in the sense that these genes which is transferred from HFR cells will attach to this bacterial cells, so bacterial chromosome, so it is called the recombinant one. So next is the matching of the H prime with the H minus. So what happens is that the HFR cells, what happens is there will be the deintegration. So deintegrations of the F plasmids, and it will take some of the parts of the bacterial chromosomes. So whenever we keep them S prime and the S minus to do the conjugations, then this DNA, so these plasmids will be transferred into here, just like this, and all these plasmids along with the bacterial chromosomes will be transmit here, just like here. And what happens is that the matching of the F prime and the F minus results in the F minus to transfer into the H prime. So this is the result. So what actually happens is that if we make the bacteria to react with the or to mate with the S plus, if we keep the female bacteria to mate with the F plus bacteria, then it will form the F plus. If it will mate with the HFR, it will be HFR, and if it mate with the F prime, it will be the F prime. So it is very easy.